of you to this very important webinar. This is the third session in the IAS series of seminars in nanotechnology. Our guest today is Dr. Alaaldin Alkilany. He is an associate professor of pharmaceutical nanotechnology in the pharmacy school at the University of Jordan and the director of Accreditation and Quality Assurance Center at the same university. Dr. Alkilany obtained his bachelor degree in pharmacy from Jordan University of Science and Technology. Afterwards, Dr. Alkilany joined Hikma Pharmaceutical as a formulation scientist in research and development department. Dr. Alkilany participated in the development and registration of more than 20 pharmaceutical products in the local and international markets. After obtaining his PhD, Dr. Alkilany joined the Medical College of Georgia, USA, as a postdoctoral research fellow, where he received a National Institute of Health Fellowship to perform multidisciplinary research on the use of gold nanoparticles to target vascular tissue in animal models. Currently, Dr. Alkilany research focuses on laser-based cancer treatment using plasmonic nanomaterials, developing a sensitive plasmonic nanotechnologies to probe and detect biological and cancer markers, discovery of novel pharmaceutical nanocarriers towards enhanced therapeutics. We are very pleased to have you, Dr. Alkilany, on board. Please go ahead.
Thank you very much, Professor Abdullah, for this nice introduction and for this also invitation. It's my truly pleasure to be with you, and I will start to share my slides. Please confirm to me that you see it good and you hear me good. The voice, everything is fine, right?
Yeah, go ahead, please.
بسم الله الرحمن الرحيم والصلاة والسلام على أشرف المرسلين محمد بن عبد الله الصادق الأمين. اللهم علمنا علما نافعا ننفع به وننتفع به واجعله حجة لنا لا حجة علينا. So today I will tell you a little bit about this emerging field called nanotechnology, and due to the broadness of my audience, I'll try to be as brief as I can, as non-technical as I can, and I hope I will deliver a summary or message about nanotechnology in this 45 minutes. So let's start with back in history. Humans and many civilizations have been fascinated by big things, like by sun, by moon, by stars, by big buildings, big statues, and things like that. If we go back to الآية الكريمة المعروفة فلما رأى الشمس بازغة قال هذا ربي هذا أكبر. So Ibrahim alayhi salam was fascinated by the sun more than the moon, because it's larger, and it is mentioned also in the Quran about the Iram Dhat al-Imad, just to show how this civilization was really great. And the Egyptians are always get proud of their pyramids, and they're till now standing big buildings, architectures, which just deliver a message for the humanities all the time that a great nation was living there. The same thing in Jordan. If you see this picture now, which which is of Petra, a huge carved city in the stone, also highlight and strengthen the same motion here, or the same notation, that humans and civilization have been really facilitated by big things. But at the same time, there is other world, which is the small world or the smaller world. also attracted the attention of humans, especially physicists, scientists, chemists, and biologists, because after the invention of microscope, a lot of interest was dedicated to understand the building blocks of material, like the atoms, the atomic structures, the electrons, and the molecules, and a lot of chemistry and interaction at that small world. This picture to the left shows gold, but two forms of gold. A bulk gold, this is a piece of gold which is 18 centimeters, and we have like atomic gold. This is one atom of gold. So this is the big world, the bulk world of matter, of gold, and this is the atomic level. Actually, this picture is part of a publication in Science just 4 days ago, showing the highest spatial resolution of seeing atoms by the high resolution or the fancy electron microscope human was able to build. So this small world actually fascinated a lot of scientists to unveil the fundamentals of it, to understand it more, to try to manipulate that, and to see what is the property of materials at that level of dimension. And here we cannot really miss this very important lecture in 1952 by Richard Feynman, who is an MIT professor. He was a theoretical physicist and Nobel laureate. He was predicting the nanotechnology in advance. This is his sentence: when we go down in size in any material, physics should be different. There is plenty of room at the bottom. So this was a prediction based on a theoretical calculation and imagination that if you go down in material and you decrease the dimension of the material to a level that you will now see in new physics, in new chemistry, in new biology, new interaction, in new properties, in new applications. And this prediction nowadays has been confirmed by the word called nanotechnology. So between this bulk material of gold and atomic material of gold, there is other level of material dimension, which is the nanoscale. I mean by the nanoscale here, 10 to the power of -9 of meter. When we say nanometer, it is really very small. If you want to imagine how small is that, imagine the length scale that our nails will grow in a minute, just in a minute. So our nail is growing continuously. In a minute, the length scale of this growth is the nanoscale. So it's really, really very small, larger than the essential building block of any material, like atoms or molecules, but way smaller than the bulk itself. So this nanoparticle, which is a gold nanoparticle of diameter of 20 nanometer, contained like around 300,000 of atomic gold. So still larger than a gold atom, but way smaller than the bulk. So this is the definition of nanomaterial, which is an interface between the bulk and the building block. So it is the material with the dimensions between the bulk and the building block. So the essential question here, so why it's really important. Before that, let's just strengthen our imagination. How small are these materials? If we compare the diameter of this therapy ball to the diameter of the Earth, it is exactly the same as the diameter of gold nanoparticle to a basketball. So imagine how small is this just by knowing how small is this compared to Earth. So we're talking about really something very, very small, approaching the dimension of protein, albumin, way smaller than the cell itself or the bacteria, approaching the same size scale of virus. If we look at the COVID virus, it is around like 70 nanometer in diameter. So this is really a small building block. But why it's important, back to the very important question now. So it is a small, but okay, what is special about being small, or what is special being at the nanoscale? Okay, before we go into the details, let's just put this question. If I want to ask you to choose which car you want to select, to the left or the car to the right, I'm pretty sure all the audience will go with the SUV. It's roomy, it's bigger, you know, but always, because we think like always bigger is better, yeah. But it's not the case in every case. No, in this case, the smaller one make it like safe, and it was away from this tragedy. And this escape was due to the size itself. This is exactly what happened when we inject nanoparticle into blood targeting the tumor. If you see this cartoon show like a blood vessel, and this is a cancer or a tumor region. In the tumor region here, this line, which is the endothelium, these are like endothelial cells flooring the blood vessels. They are intact. They are making like no gaps between each other, just to preserve the content of the blood, just to prevent all the cells or the proteins to escape to the extra matrix. But at the tumor region, this endothelium is disrupted, as you see here in this picture. And we can see it's not intact anymore like it should be in the normal tissue. It's now like more porous, more leaky, allow penetration or escaping of this nanoparticle into the cancer tissue and accumulate selectively in this region, as we see here. This is called the passive enhanced retention and permeation effect. In other words, it is the passive targeting to the tumor or to the cancer tissue. And this is the only reason why this nanoparticle will end up in the cancer region, not in the normal region selectively, is just the size, because it can make a benefit of the leaky nature of the blood vessels in the cancer region itself. This is very, very like in a very close resemblance to the example I just showed you regarding the cars, where smaller is better in this case. So this is like one of the biggest advantage of nanotechnology in biology or in medicine, that we can make the drug smarter, not going everywhere. As we know, cancer like medicine or anti-cancer therapeutics, they are blind. Upon like injection or upon like uptake, they're gonna go everywhere. So they're gonna kill everywhere because they are killers. They are cytotoxic moieties. So they are. We are designing anti-cancer to kill cells. But the problem that this drug will go everywhere and will kill the cancer cells, the good cells, and we don't have real selectivity where to kill and where not to kill. But in this case, the nanoparticle can be accumulated selectively in the tumor or in the cancer region, and we can ensure selectivity and targetability. And this is a wonderful and cannot be really achieved without making utilization of the site itself. So this is one example of how the nanoparticle is really, really good and have like unique properties that can be employed for various applications. Let me give you another example. Now, if material has like centimeter times centimeter times centimeter cube, and you did reduce this by milling, for example, into nanoparticulates, generating like many nanoparticles out of this building block. If you calculate the total surface area now for the finer powder or the finer particle, now the surface area is way much higher than the original building block. The increase in surface area is very important because it can just improve the dissolution of material. And this is a big challenge for pharmaceuticals. For pharmaceuticals, many drugs are really exhibit poo solubility. Just by enhancing that surface area, you can ensure that the drug will dissolve as it should, and it will provide like higher bioavailability in the tissue. And the higher surface area also allow us to decorate these nanoparticles with multi-modalities or multi-entities. We can like put imaging compounds, therapeutics, specific targeting moieties, cell-penetrating agents, all of these at the same nanoparticle, generating what we call multifunctional nanoparticle that can target a specific cell, can be used for imaging and therapeutic at the same time. So this cannot be done without going at the nano scale. So this is a second argument about the importance of nanotechnology, or why small is really important. But the two examples I gave so far related to biology, let's just give some examples related to physics and chemistry, explaining the importance of nanotechnology. If we see the melting temperature of gold as function of the particle dimension, particle radius in this curve, we can see that the melting temperature for bulk gold approaching more than 1000 Celsius degree, where if you go down in size now, you start to change the melting point. And we know melting point is a physical constant. It should stay constant. But at the nanoscale, it's even the constant physics is changing, as Feynman was predicting in my first slide. So this change in physics, and also there is a change in chemistry, because as we know, gold is inert, will not react, it's stable. And this is why we see it from thousands and thousands and thousands of years in the ground, right? Without any change, without any oxidation, for example, like other metal. But gold is inert and noble. But at the nanoscale, the inertness in chemistry is not any, it's not anymore true. In fact, nano gold can be used as catalyst, not just reactive by itself. It can also catalyze other reactions. So there is a big difference in chemistry and physics also at the nanoscale when we go down in size. And this is what makes nanotechnology revolutionary. Why? Because the revolution here comes from that each element in the periodic table, if you like prepare the nano counterpart of it, you can witness new physics, new chemistry, and new biology. And this is really wonderful, because imagine if we discover just a new element we add it to this periodic table. This is going to be a huge discovery, just one element. Now, what we are doing, we are not discovering a new element. No, we're discovering a new properties of every single element at the nanoscale. It's look like we can duplicate the periodic table. And this is why nanotechnology is revolutionary. Okay, let me give you an example about how physics is different. If you consider gold, I'm focusing on gold because it is my research. Because when we talk about nanotechnology, it's a broad spectrum of particles, of materials. So you have like nanosilicon, nano gold, nano silver, nano oxides, nano organics, liposomes, many players here. So my specialty is gold, so I'm trying just to use as many as I can from my field here. Okay, how physics is different at the nanoscale? Let's consider the optical property of gold. As we see, gold is golden in color. And why it's golden? Because it's simply reflect light. But if we make gold nanoparticles, now these nanoparticles, they absorb light, they scatter light. And in physics, absorption, scattering, and reflection are totally different physics. Different interaction between photon and electron at the surface of the material. As you see, these vials, which was prepared in my lab at the University of Jordan, all of these are just gold nanoparticles. These colors are not from dyes we are add to these vials. They are not chromophores. No, these are, all these vials are just gold nanoparticle with different size, with different shapes. The same material exactly of this gold. But because they are small, now they are absorb or scatter fraction of light, leaving the other fraction. And this explains why these vials look red or violet or blue or brown. It's fascinating that we can prepare now red gold or blue gold or green gold. If we go like 100 years ago, nobody will believe us that these are just gold nanoparticles suspended in water only, and they look colorful because we know gold is golden or yellow in color. And physicists now, they can model actually all these interactions with light and the absorption and the extension spectra, and you can even predict the absorption spectra or the optical extension spectra if you know the size and the shape of the nanoparticle. So we have new optical properties. And what is the importance of these optical properties, as I will show you? It is the fundamental or the basis of imaging applications, photothermal applications to treat cancers, sensing applications to look up for analytes in any system. I will elaborate on that. As we know, gold just reflects light, but gold at the nanoscale now absorb light. And if you zip these nanoparticles with lasers, with the proper light source, you will excite electrons in the conduction band of this metal. And these excited electrons, they will decay their energy finally in the form of heat around the nanoparticle. Because you know, you're bumping energy into the system, electrons will get oscillated, will get like kinetic energy, and this kinetic energy will decay, and this decay will be in a form of local heat. So if you decorate the nanoparticles with a smart ligand that can target the cancer tissue, and you successfully allow more accumulation of these nanorods into the tumor tissue, and after that you shine it with light, so you generate a local heat which can be employed to kill cancer cells, bacteria, any unwanted organism of interest. A good point here I need to mention that the optical property can be tailored or can be manipulated just by controlling the size. As we see here, we can just go from red all the way to brown just by elongating the spherical gold nanoparticle to spheroid and then to gold nanorods. And this is really interesting. Why? Because gold nanorods absorb at 800 nanometers. At this wavelength, our body is transparent to light. So it will penetrate deeper into the tissue to reach the gold nanorods and to generate the heat and kill the cancer. This cannot be done using spherical gold nanoparticles, which was prepared in 18, like maybe 90 by Faraday. This was the first gold nanoparticle prepared, and at that time he was not really termed that nanoparticle, and he was not aware about the concept of nanotechnology and many characterization of his preparation. But this like simple old sphere will not support this photothermal therapy simply because they are absorbed at 520, 550, where our body is not transparent. But at 800 in the near infrared, more photon will make it deeper into the tissue and reaching the gold nanorods, which will absorb that, turn it to heat to kill cancer. So there is a very different optical property between bulk and nano. And this what allow us to use these nanoparticles as phototherapeutic agents to treat cancers and bacteria or other viruses. Okay, so this is an example about how physics is different. And here, this is from my own work. We can use these gold nanorods if we functionalize it with a targeting moiety to kill cancer, as you can see here. and we can see these nanoparticles inside cells clearly using electron microscope, as you see in this picture. These nanoparticles were engulfed or endocytosed by cells, and after that, we can zip it with the proper laser just to induce killing for the cancer cells. And other groups, many other groups now, this is the work of a group from MIT. They can show the use of the photothermal capability of gold nanorods in vivo, ex vivo, and now in clinical trials. As you see here, a complete irradiation of the tumor can be achieved using gold and the proper laser wavelength, and save the life of these animals. Okay, so these like properties cannot be there without going small in size to the nanoscale. These examples were about gold, and I'm selecting here another example regarding the cadmium, which is like a metal with a silver color, as you see here. And if you manage to prepare these the same material with nanoscale size, like 2 nanometer to 8 nanometer, you can witness a new phenomena, which is the fluorescence. So if you remember the gold, we were talking about absorption and scattering. Now we are talking about fluorescence. It's totally different like optical physics. And this fluorescence or new fluorophores can be now used for in vivo imaging and detection, and they are way brighter than classical fluorophores, and more importantly, they are more photostable, and they can also prevent many artifacts like such as desorption of the fluorophores from the surface. I don't want to elaborate on the fundamentals too much, because maybe my audience is just broad. So I'm trying just to be as simple as I can. Okay, very important question. Like, okay, these are very good examples about nanoparticles and why they are important and how they are different and the different applications that we can use these nanoparticles for. But always there is a big question. Nanomedicine, is it real or not? Let's go and look from the market now, not from the research papers or from lectures only. Just look at the market. Do we have like nanotechnology-based medicine? The answer absolutely yes, and this is from 1995. This is the first FDA-approved nanomedicine, the Doxil, to deliver doxorubicin, which is anti-cancer using liposomal nano delivery platform. This is a transmission electron microscope, show a crystal of doxorubicin inside a single liposome. And this liposome will enjoy the enhanced retention permeation effect, as I demonstrated in the first slide, go more selectively to the cancer region compared to the normal and healthy region. After this example, or the first nanotechnology, or before I mention more, I want to just remind my audience that this delivery platform, okay, which was approved in 1995, the liposome, is very similar to the platform we are using now in the COVID vaccine. The COVID vaccine now, not all of that, but the mRNA-based technology, we cannot deliver mRNA to the tissue naked because it's not stable. It will be chopped and degraded before it's reached the targeted cell. But how we deliver now the COVID vaccine is just we engulf or we wrap up this mRNA inside the liposome or lipidic nanocarriers, and this will allow this mRNA to get inside the cell and then get translations like reactions and presentation of the antigen and start the cascade of the immunity buildup. So this is very important to highlight that the vaccine, the mRNA vaccine, will not be reality without a nano carrier that will act as a shuttle to deliver it and maintain its stability to reach the targeted cell and start its work intracellular. After the doxorubicin, the market witnessed a lot of other FDA approvals for many other modalities. As we see here, this is the Amphotericin B liposomal formulation. This is the Abraxane, which is like a protein-bound anti-cancer. And we have also, we cannot miss the Ferrahim, which is iron oxide nanoparticle for anemia treatment. And it is also magnetic. It is also magnetic, so used to support MRI imaging. Also, this is another example about the use of nanotechnology in reality or from the market. What you see here is the pregnancy test. I'm pretty sure most of my audience know that, and it is one of the cheapest sensing platforms available in every single country around the world. You can get it from the pharmacy to test the pregnancy status, and it is based on lateral flow immunoassays that you deposit droplets of the analyte of the sample containing the analyte, like for example the urine, and this is going to flow laterally. And there is a gold nanoparticle here functionalized with antibody that recognize this analyte. And if the analyte is there, this will end up in a sandwich like architecture as we see here, and assembly of the gold nanoparticle into a straight line. And this straight line, which will give you the positive result that the analyte is here or not. And the red line reminds us about the color of the gold suspension, which I showed you in the first or second slide, the spherical gold nanoparticle, the red one. So if you go to the anatomy of this testing platform, these colors are coming from gold nanoparticles. And why we are using gold nanoparticles in this sensing platform, because their optical extension are way higher than any chromophore we know so far, at least 100,000 times, 10 to the power of 5. So you need a little bit of gold to see the optical response or the color. But if you want at the same concentration, you want to use a coloring agent or a dye, you need way higher amount. So this is also a good example showing how nanotechnology in a real use in the market. The same technology, that technology was really old. We know these testing platforms, they are in the market maybe since 50 years old. And the same approach was used to detect the COVID-19 using the lateral flow rapid assay geometry and looking for the virus itself or for part of the virus. Okay, so I wish I was successful in delivering so far a general definition of nanoparticle, nanotechnology, why it's important, the new physiochemical properties and biological properties of material at the nanoscale, and a little bit about the applications of these nanomaterials, and some examples from the markets highlighting the presence of these nano platforms in our use, from sensing to therapeutics to cancer treatment. So, to my audience, this is the message before we start with the other parts, that we cannot really cover the story of nanotechnology in one lecture. It is really big field now with a lot of specialties and with a lot of details. So I was preparing this talk just to deliver a little bit of the fundamentals. Before I go, I cannot miss this part, which is nanotechnology in nature. I like it personally, and here I need to remind my audience for this ayah, which is صنع الله الذي أتقن كل شيء إنه خبير بما تفعلون. So صنع Allah, this is what Allah made. As we see, this is the amazing wings. These colors in this butterfly and in many other insects, actually. It's not just a butterfly. All these colors we see, the beautiful colors, it's not chromophores, it's not a real dye. No, it is a nano features. The material of the wing itself has this design of architecture and featuring, like the periodicity of these features will dictate the color you see. If you manage to make these features away from each other, like even further or closer, so you can now generate red, maybe green, blue. So all these colors are just architecture features at the nanoscale in the wing of the insect itself, and not a true color. We learned from that, from the nature, and we were able to assemble these aluminum oxide nanoparticles in a controlled manner, make them close to each other or away from each other, and now we can control the optical properties. We can make it like yellow, red, blue, to generate like nanoparticle liquid crystal displays and optical filters. Another example from the nature, which is a really great example, is the gecko which is a natural nano glue. If we ask ourselves why this gecko can really just stick to the surfaces in general, to the wall or wherever, stick very, very smartly. What's going on there? If you do like a microscope imaging for the toe from the interior part, you will see all these features. All these features are at the nanoscale, and the high surface area generated by these nano fibers, they are responsible for the high binding with the surface through the hydrophobic interaction. And we learned from that to design like smart glue or super adhesive glue without a real polymer in it or a real glue in it. And this was an NSF funded project where we learned from the nano features in the gecko to build what is called now the Nano Magic Tape, and it is now in the market. You can order it from Amazon, produced by 3M and other companies. And what they did, they utilized carbon nanotubes in a resemblance to the gecko toe. And you see, maybe this is a PhD student, like testing hair, like preparation. And what you need to do is just stick it to the surface, and when you pull it down, you generate more contact area with the surface, and the gluing effect will be apparent. If you take it in this direction, you can just take it from the surface. You can peel it off. So, and you can use it again and again and again. It's not like the usual or the normal classical glue. So this is like a good example about how we can learn nanotechnology from nature. And the third one is the super hydrophobic Lotus leaf. This is the Lotus leaf. If we go to any pond or maybe lake, small lake, we see these like beautiful green leaves. And this leaf repel the water really excellently. And why is that? Because we have a lot of nano features in their surfaces. It's not enough to be hydrophobic to repel water and induce a high contact angle, as we see in this picture. You need high hydrophobicity and at the same time nano features in the surface to maximize the hydrophobicity and make the water droplet just hate the surface completely, because the water is hydrophilic and love hydrophilic in nature. And the surface now is hydrophobic, doesn't love water. So the water droplet tries to minimize its contact with the surface and to adapt a spherical shape, just not spreading all over around the surface. We learned from this nano feature-based super hydrophobicity to now generate more material which can be super hydrophobic and repel water, as we can see now. I'm pretty sure many of us now saw these textiles in the market from Adidas, from Nike. Many companies now are doing that. That you can spill juices or water or any liquid on the textile, and you end up with nothing wetting that textile. And the same phenomena was used to prepare like self-cleaning glasses, where the droplet of the water will just keep rolling and taking all the dust with it, and not just sticking and spreading and evaporating and leaving the debris and residues on the surface itself. And we used also that to fabricate like smart masks for the COVID pandemic fight, or during this fight. The outer layer will be super hydrophobic, preventing the first step, whereas the aerosol or the droplets will not even stick to the surface itself, will get repelled. So yeah, there is a lot of like natural also nanotechnology platforms. We try to understand it, and thanks to advance in microscopy and in analytical chemistry and in physics and in chemistry, which allow us to understand these systems very well, and not just to understand it, to make it, and to control our synthesis, even to control the size, to control the shape, to control the surface chemistry, and upon that to open a lot of applications in different like disciplines like biology, medicine, pharmacy, and engineering. So nanotechnology is intersection really between many fields, and naturally it's a multidisciplinary field. It's really interesting, and it's really worth like more attention in our like countries, in our universities, and in our industries, to try to catch up now the global interest and the global investment in this emerging field or emerging theme. So, and before I leave, I would like maybe to thank, first of all, the Islamic World Academy of Science. Really, I truly appreciate your invitation, and really it's my honor to be a speaker today through your platform. And also I would like to thank Jordan University, where I do work, for their continuous support through like funding and other forms of support, and also the Deanship of Academic of Science and Research at the University of Jordan. I cannot miss also DAD, the German Institution for Academic Exchange Services, and also the Alexander von Humboldt. I was the recipient of this award for 2 years in Hamburg. Also, I want to really thank a lot of people here, but maybe I particularly like 3 professors with a close ties and close collaboration and continuous one, which is Professor Catherine Murphy from University of Illinois, which I'm visiting now, and Professor Wolfgang Barak from University of Hamburg, Germany, Professor Murtaza Mahmoudi also from Michigan University in the USA. And all my students, without them, nothing can be done from my own research. And thank you very much for my audience and for the panel organizers.
Thank you very much, Dr. Alkilany. Really was very interesting with the huge body of information that we get this morning.
Thank you so much, Dr. Abdullah. I wish I tried to make it very simple and very like non-technical, because this is actually my big challenge whenever I deliver talk to like broad spectrum of audience. And yeah, thank you so much.
Yeah, you did excellent job, Dr. Alkilany, I tell you. Thank you very much. Now we will open the floor for discussion and questions. And I have Dr. Azhar with me, and Dr. Shanshar probably sent something as well.
I have a comment, please.
Yeah, you have a comment.
A short comment, yes.
Yeah, please go ahead.
First, let me thank Professor Alaaldin for his nice talk. بارك الله فيه والله يوفقه إن شاء الله دائما.
الله يكرمك.
We look forward for more achievements by him. And my comment is that people always talk about Feynman's prediction about the possibility of new chemistry or new physics for the sub-nano microsystems. Now, this was a hypothesis which had to be proved. Dr. Alaaldin Alkilany.
Yes, Doctor. معك.
Dr. Alaaldin, تسمعني؟
معك دكتور. Yes, 100%.
Dr. Alaaldin, this was a hypothesis, but it should have been proved either by a mathematical approach or by an experimental work. Now, as much as I know, this experimental work was done in the 70s by Herbert Gleiter, who studied the electron emission microscopic pictures for molten alloys and molten gold metals under the super cooled conditions. Now, from his work, the so-called the boundary grain and boundary concept was developed, and from that, of course, he generated the whole business afterwards for a new world of nanoscience and nanotechnology. Now, people seem to disconsider this fact. Herbert Gleiter has not been mentioned at all, whenever a talk is being done about the nanosciences. Instead of that, Feynman's words, which are hypothetical, they are not proven by a mathematical approach, non-quantum mechanical approach, and he did not do any experiment like Herbert Gleiter. Gleiter did the experiment for that, and he has his great the boundary concept, grain and boundary concept, which he followed for years, and then he developed the whole business of the nanotechnology. I think this should not be avoided at all. I feel sorry for this fact, and for the sake of scientific الأمانة العلمية, one should always be conceptual and consider these facts exactly.
أعطيك رأيي دكتور مثنى. أعطيك رأيي. سامعني دكتور مثنى.
Would you, Dr. Alkilany, would you speak in English? So that
I'm sorry, Dr. Abdullah. Yes, Dr. Muthana, thank you very much for this comment. Actually, this is like very complicated thing, as you know, to give the credit for the right person. Well, this is why the ayah is saying, عند الله تجتمع الخصوم. This is very difficult to assign, who is the father of this field, who is the pioneer in this field, who is the initiator of this field. I have different opinion than yours, Dr. Muthana, if you allow me. Feynman is a theoretical physicist. He's a theoretical physicist. He's a pure theoretical physicist. He's doing nothing with experimental physics. So his prediction, he was doing a prediction. It's not a hypothesis and need like an experimental support. No, he was doing like prediction more than experimental. But if I want to give a credit for someone, I will not give it for Feynman nor for the other scientists you mentioned. Maybe if we go back to the 18th century, Faraday, he is the father of nanotechnology, the real one. The first one who prepared colloidal gold nanoparticle is Faraday, and you can see it intrinsically in London Museum till now. Till now, it is. If you look up the website of, I didn't see it by my eye, but if you look up the website of London Museum, where you can see their thing, you can see the Faraday colloidal gold, the red one, exactly as we prepare every day in my lab. At that time, he was not called it nanoparticle, he was called it colloidal system. He was not really interested in their biological applications, but he was really reporting that this system is totally different than any true system. There is a sedimentation effect, there is a settling effect, there is a redispersion effect, many things he was describing. And very interestingly, also his notebook, also in the museum itself, you can see it also online. You know, so maybe if I want to name someone as the father of nanotechnology, I will call it Faraday. This is an opinion, and also it's also still an opinion. You know, it's open. Yes.
Allow me a short comment. You can go back to the Damascus sword, which is known to be
yes.
Of nano iron, but this is not the way. This is not the way. These things should be based on strong conceptual basis. And Herbert later did that. He did the electron emission microscopic pictures of the supercooled metal alloys or supercooled, I think, even gold metal, and he was able to describe the system as the boundary and the what's the other name, Habbat, and then he developed the whole concept with the time. Now Faraday did that, but this is the same as the Damascus sword. Nobody knows what he has done.
Not the same, Dr. Muthanna. No, in Damascus, we discovered that there is nano features in that sword, as we discovered that there is a lot of nano features in the church windows in many churches, right? We did discover that, but no, Faraday was doing scientific. Faraday is the best physicist, chemist known to history. But he was doing experiments and laws. Faraday law is very known. Yes.
Did he talk about nanosystems?
Yes. You will be amazed. Actually, I went through his notebook when I had free time, and you will be amazed how Faraday was knowing the nanotechnology in the 18th century. It is really amazing. I can't share with you like many materials because we did like I can share with you like a lot of website to his notebook because it is online. You can see the picture of his notebook online, you know, and his preparation is fascinating also, the gold nanoparticle. And he was describing, do you know the Faraday driving force? He was trying to see the light material interaction when we go down in the thickness of a metal layer. And the first step, the first stage of his research was to hammer like a gold piece, to make it thinner and thinner and thinner and thinner. And he realized he cannot really reach a level where light can just go through it or change anything. So he started like to synthesize the gold and make it settle down to assemble into like a packed monolayer of colloidal nanoparticle, which can form like the thinnest leaf he can generate. So he was aware about all this, but definitely not like our knowledge now. We can now see a gold atom in a lattice of gold nanoparticle. We can like, as I mentioned in my talk 4 days ago in the recent issue of the journal Science, we can see a single atom. Like seriously, we can see just a single atom. This is the highest spatial resolution obtained so far. I agree with you, Dr. Muthanna. There's many scientists like really deserve the credit, definitely. But Feynman, it's not my opinion. I'm pretty sure you know that you can see the same argument in many like publications in the like most prestigious journals about the contribution of Feynman. But it's I agree with you. It's many people also like have contribution and yeah, it's the field will grow definitely by the contribution of money from the old people and from the current people.
Okay, thank you, Dr. Shanshal and Dr. Ala, for this nice debate about the history. But let's give the floor for some more comments in the core of the thing. I think
Yes.
Dr. Azhar has something to say in this regard, because he sent me some questions and I don't know whether he wants to ask them. You are muted, Dr. Azhar. Dr. You are muted.
I hear him.
Yeah, okay, but you are muted. You would like
Hello?
to ask the questions, or should I ask them?
You ask, because I will comment then.
Okay. Dr. Kilani, here are 6 questions for you from Dr. Azhar. Question number 1. Does nature itself use these nanostructures in its pharmaceutical operations?
Excuse me, Dr. Abdullah, again?
Does nature itself use these nanostructures in its pharmaceutical operations?
It's biological, you mean? Biological operations.
Okay, go ahead.
Yeah, Dr. Abdullah, definitely. Like, yes, there is many nano-based biological systems. If we look for the albumin is a nanoparticulate. Albumin, which is the most abundant like protein in the blood, it's a nanoparticle, and this is the reason why we use it in Abraxane. We attach drugs to the albumin to deliver anti-cancer. If we look how the kidney filtrates, our blood. The kidney, if you look about the pore size of the kidney, it's around the 50 nanometers. So it's a nanofilter. The histone, the histone system is a nanoparticulate. The DNA is a nanoparticulate. We can now, the first motor we can prepare, it's based on a DNA. The DNA assembly can be used in many nanotechnology based things. So definitely there is in the nature there is way more nanotechnology examples than we did as chemists, and even way more beautiful. And this is why I put a couple of like slides just to describe subhanallah khalqullah fi hadha al-itijah, how Allah created all these systems just on a beautifully. Yes, the question definitely we have a lot of nanoscale like operations or materials or events happening in biology. Definitely, yes.
Question number 2. Why the nanostructures have to be preferably anisotropic to be effective pharmautically?
Well, doctor, in all the pharmaceutical nanoparticles in the clinic so far, they are isotropic and not anisotropic. A liposome is isotropic; it's spherical. The Abraxane is albumin protein is spherical. The pregnancy test I showed you is isotropical, not anisotropical. But I agree with you that a lot of feature will show up when you do anisotropic chemistry. Definitely. For example, if you prepare a gold nano rod, which is my specialty, by the way, the gold nano rod, now we can support the NIR optical absorption and scattering, which allow deeper penetration to the tissue for more successful therapeutics. But this will not be beneficial if we use gold sphere. So you don't need to go anisotropic, and all the examples in the market are isotropic. But I agree with you, there is a lot of feature will show up only and only if you manage to make to break the symmetry and make it anisotropic. Yes, doctor.
Okay, question number 3. Are the organic nanostructures pharmaceutically more effective than inorganic ones for our carbon-based dyes?
Well, doctor, I cannot like answer sharply here, like this is more effective or not. Definitely every platform has its own pros and cons, advantage and disadvantage. So for pharmaceutical delivery specifically, the inorganic is not really preferred due to challenge related to elimination, excretion, and maybe accumulation and toxicity. So we don't know a lot about these things. So we cannot, like, for example, treat the Alzheimer just by injecting gold nanoparticle into the brain. It will not get cleared, it will not be eliminated or metabolized, or we don't know after 10 years what will happen, you know, with the gold inside the neuronal mass. But if you want to do like the pregnancy assay, it is the best thing to do is just gold, for example. If you want to do like photothermal therapeutics using localized tumor, specific type of tumor for specific settings, maybe gold is better than carbonaceous material. But in other cases, maybe protein-based is the best. Other cases, maybe the liposomal rationale is the best. So it depends on the application, on the settings to rationalize or to design the best nano platform you will go with.
Question number 4, does the surface plasma in metallic nanostructures, such as gold, has any pharmaceutical role beyond being the source of rainbow of colors?
Oh yeah, Doctor, definitely. Thank you for this question because this is very important. Yes, the LSPR or localized surface plasmon resonance or effect, it is the basis of all other applications. The photothermal effect is related to that closely, because at the end of the day, it is a photon-electron interaction between light and matter. And the surface enhanced Raman spectroscopy, which is an approach, a very strong analytical approach now toward a single molecule detection, is based on the LSPR also. The imaging also, there is LSPR-based imaging. Definitely, there is a lot of like pharmaceutical and biological application related to imaging, sensing, and therapy based on the LSPR. And this is not surprising because the LSPR, by origin, it is electron-photon interaction, and all these applications indeed rise from that.
Any possible intrinsic harmful pharmaceutical effect of these nanostructures in the living body?
Well, definitely nothing is 100% safe. If we swallow like 10 pills of paracetamol, we'll end up with kidney failure. So nothing is like safe. It depends on the dose, on the therapeutic threshold, in many aspects. So we cannot say it's safe or it's not safe unless we came to a point where we know the therapeutic behavior of the gold, and we determine the therapeutic dose we need, and at that like level, determine the toxicity of this therapeutic level. But in short, gold is promising because it's benign in nature. It is benign. Remember, there is many drugs based on gold salts, right? The gold therapeutics, for example, it's based on gold salt. We used to put like gold teeth and gold in knees and gold. So gold is benign intrinsically, and accumulated like research bodies support that it has like really minimal toxicity. But at the same time, it depends on where you are injecting and how much you are injecting and for how long you are injecting. For example, as I mentioned, like gold to treat Alzheimer is not like gold, for example, to treat a localized tumor here and with external light. It's going to be totally different settings and different like judgment for the toxicity. But I agree, toxicity is a challenge, need to be addressed. We need to know more about that. It is something we need to address, but in my opinion, it's not an obstacle that is gonna stop this like inventions from being reality.
The last question from Dr. Asghar: will you consider the acupuncture work a pharmaceutical operation. This is away from nanotechnology, I think.
Yeah, it is a way, I think. To be honest, Dr. Asghar, I don't have like a lot of experience in it, just a very superficial knowledge about what it is.
Okay.
Thank you, Dr. Asghar, for yes.
We now have Dr. Hamad to ask his question. Dr. Hamad, please.
I have one question.
Dr. Hamad.
Assalamu alaikum and thank you so much.
Wa alaikum assalam.
Ahmad Arshad, a faculty member at Lahore Garrison University, Pakistan. First of all, thank you so much, Professor, for this wonderful talk. So I have a question regarding the synthesis or the production of metallic nanoparticles using photoirradiations. You mentioned in detail that how these nanoscaled metallic nanoparticles, specifically gold nanoparticles, behave differently as compared to their bulk material. So I just wanted to know if you could please enlighten the mechanism behind the synthesis of these metallic nanoparticles when we expose these raw materials, for instance, the salt of silver or gold, and some reducing agent, for instance, some biological material, let's suppose plant extract or something else. There are many reports, but I wonder how these simple radiations, whether solar light or LED lights, behave as an inducer to that the sorts of those metals, and then they convert into the nanoparticles.
Yeah, okay. Thank you so much, Dr. Hamad. I tried to skip the synthesis part, because as I mentioned that I will be broad, But yeah, since it is your question, thank you so much for that, because this will make it an opportunity to share a little bit of synthetic details with the audience. Specifically for metallic nanoparticles, the synthesis, as you know, can be classified into two major routes, which we can go from the atomic level to the nanoparticles, which is the bottom up approach, and the other approach, we can bring the piece of gold, the bulk itself, and just chop it into smaller pieces, which is like the top down approach. Okay, so we can generate nanoparticle two ways. In each class, there is many subclasses also. Okay, for gold, specifically for gold and silver, the photo radiation you mentioned, I think you mean the photo radiation of metallic salt in solution, right? Not the lithography by definition. Not you are irradiating the metal itself and you generating like nanoparticle out of that. Not the lithography.
Yes.
You are talking about salt dissolved in water, for example, and you just shine it with light, and this light will reduce the gold or will help reducing the gold to generate the nanoparticles. This is your question, right? Yeah. So for gold and for silver and for other metallic systems, it is a simple reduction system. So we have the gold Au plus 3, for example, or the silver Ag plus 1, or other metals M plus X, okay? And you put the reducing agent. So the reducing agent could be a chemical molecule like ascorbic acid, sodium borohydride, hydroquinones, many reducing agents there are around, okay, to induce the reduction or the redox chemistry. Supply these Au plus 3 or Ag plus with electrons to make them Ag0 or Au0, which is the noble gold or noble silver, which will find itself in water, not supported with high interfacial tension. The only way for them is just to stick to each other and form the nanoprecipitation or the nanocolloid in the presence of a surfactant. Okay, they will come adsorb to decrease the surface energy and make it like a stable colloidal system. So this chemistry is the same chemistry Faraday was using, by the way. He was dissolving gold piece in aqua region, okay, and then reduce that, okay, with black phosphorus. This is his recipe, by the way, in the 18th century, okay. So Dr. Musanna, he was doing experiments, and he was like optimizing the reducing like concentration and the goal to reducing. The same paper in Nature, 1972, published by Friends. I'm pretty sure, Dr. Hamad, you know this one, which is the Friends method, which is simply the gold salt with ascorbic acid, which we got it from orange or whatever, and you boil it. So the ascorbic acid will reduce the gold and will act as a reducing agent. So for the photoradiation, the light itself can induce many like redox chemistry, right? So the light itself can induce reduction to the gold salt, or can reduce like a compound which you are add to the solution, and this will act as the reducing on an agent. So there is like many routes of synthesis, and this all what I described in brief is just for gold or for noble metals. If we talking about, like, for example, how you prepare like liposomes, it's totally different. How you prepare protein-based nano platforms is totally different. Carbon nanotubes is totally different. So it depends on the nanoparticle, and in general there is two ways. If you go like bottom up or top down, and for gold, like for example, the gold nanorods I'm preparing, okay, in my lab I use with chemistry. We don't use any light to radiate the salt or lithography. No, we use chemistry, okay, to manipulate the location of the gold particles in the lattice and make it the elongation in just one direction, okay. So this is just like employment of the chemistry itself to do a nice job in the synthesis. If you want, we can elaborate more on that, but I hope that I answered the question.
Thank you so much, Professor. Thank you.
Thank you.
Any more questions from the audience?
Dr.
Yes.
Dr. Abdullah.
Hello.
Dr. Shanshar, تفضل. Go ahead. Dr. Shanshar.
Dr. Alaaldin, I think we cannot consider Faraday's work for this subject, because at that time nothing was known about the atomic structure. No Bohr, no Sommerfeld, no Max Planck, no quantization rules, no Heisenberg. I think it was a type of alchemy, just like the discovery, just like the discovery of phosphorus.
Yeah, Dr. Musanna, why Dr. Musanna?
No, the discoverer of phosphorus. He took urea with charcoal and he boiled that, and then he found that this is phosphoring, and then that's it. He knew nothing about the metal, not the periodic system. Even Faraday knew nothing about that. I think this should be reconsidered.
Dr. Musanna, maybe it is not our focus, as Dr. Abdullah mentioned, the history of that, or to whom I will give the credit. But honestly, I cannot say Faraday is an alchemist. No, Faraday, he is the founder of the electromagnetics. All electromagnetics just he is the father. It's not my opinion. It is what you can see in textbooks, even not in papers. Faraday laws are the fundamentals of every electromagnetic knowledge we know by equation. It's not alchemy. So I'm not trying to defend Faraday. No, Dr. Musanna, don't understand me like that. But this is the truth. You know, Faraday is the father of all electromagnetic. And if you think about the plasmon itself as a phenomena, it is the interaction of electromagnetic wavelengths with electrons. I understand that after that, there's many researchers took it into where we are now. But this is why we call him the father, because he starts something, okay, and then many others build on that. But I cannot say that the research group 4 days ago, they published their paper in Science to see single atom, that they are the father. Okay, so they did way better than Faraday, but they are not the father. This is my opinion, but maybe I'm right. I don't know. I'm not trying to defend Faraday or other, like this is not the important thing for us, who is the father or who is the initiator of the field. I think we need maybe to focus on how we can really utilize that in our universities, in our countries, in our industries, and maybe this is the most important thing. So, but I agree with you, there is a lot of contributors around there.
No, Dr. Alaaldin, I am asking about that, not due to the knowledge of history of science, but to understand nanosciences, you have to go deep in the problem, and and find that out in terms of quantum mechanical concepts or molecular dynamic concepts, and without that, I don't seem to understand what's going on. Now, that's why when I talked about Herbert Leiter, he gave an analysis of the whole problem on the basis of the conductivity band and the change of the conductivity band.
Could we
if you allow,
interfere, please?
Dr. Abdullah, just something very important I want to share with Dr. Muthanna and the audience. Dr. Muthanna, you mentioned that you need to understand the quantization and all mechanics.
Of course.
Dr. Muthanna, the first FDA approved nanomedicine is based on liposomes, nothing to do with physics even.
Oh.
Nothing to do with physics,
Okay,
you know,
okay.
or with like or with mechanic quantum mechanics, or no, you don't need to do that. Liposome discovery, Dr. Muthanna, liposome discovery was initiated in Israel. And this is, I teach my students that Israel was the first country to produce the liposome and the nanomedicine. And it has nothing to do with quantum mechanics or quantum physics, nothing. Nanotechnology, doctor, is so broad that it's not just physics. They, without understanding any physics, they succeeded to bring that into market. I think this is what we can do and learn, how to just transform whatever knowledge is there into reality, into something which can help us as students, as researchers, and as countries. And just putting all nanotechnology in physics and the quantum mechanics, I think all the examples I showed you, I showed the products in the market, and I showed only the old examples, by the way, that most of us maybe know it. I don't want to talk about like very recent examples. All of them nothing to do with the quantum mechanics or physics. I think, Dr. Muthanna, the best thing to do is just to extract what we need really in our recipe to go further in this direction toward like translating science into industry or into like high-edge teaching or something like this. I'm sorry, Dr. Abdullah.
Oh, thank you very much for this enlightening debate, Dr. Shanshal and Dr. Alaaldin. It's really very interesting, but let's go a little bit further. I have a question for you from Dr. Kemal Yildirim from Turkey. He asked, how do you quantify nanoparticles from the pharmaceutical industry?
Thank you, Dr. Kemal. If your question is related to gold nanoparticles, my answer will be through like elemental analysis techniques, like for example, ICP mass spec, which is abbreviation for inductive coupled plasma mass spectroscopy. And thank you again for asking this question, because this is what makes also gold unique, that we can quantify it using one of the highest like sensitive and great analytical tool, which is the mass spectroscopy. Let me give you an example. If you want to quantify, if you are growing like cells in a petri dish and you inject gold nanoparticles to the growth media, after washing, proper washing, you want to quantify how much gold nanoparticle in every single cell. You can do that. You take all the cell mass, you digest that with acid, you run ICP mass spec. Simply it's a mass spec, or OEC, okay, another platform, but the mass spec is just more sensitive. So you run a mass spec looking for gold. And we know that gold in biological tissues has no background. We don't have gold inside us, right? We have calcium, we have sodium, but we don't have gold. So you have a low background, which will give you a very great sensitivity or like signal to background or signal to noise ratio. So this is one of the reasons why gold are used as probe to understand the behavior of nanoparticle in biological complex settings. Okay, so this is my answer compared, in terms of gold, if you mean gold. But my answer, if you mean in your question like the nanoparticle, it depends on the nanoparticle itself. So we look for the property of the nanoparticle, and we think what is the analytical tool suits this case. I'll give you an example. For the quantum dots, I showed you maybe the fluorescence, okay, the different colors of the quantum dots. If I'm looking for the quantum dots and I want to quantify the quantum dot level in pharmaceutical prep, I will use like a fluorescence technique if there is no interference or like interactions. If I'm looking for like the uptake of carbon nanotubes inside cells, I can label that. I can label that with a Raman tag, with a fluorescent tag, with a metal tag, okay, and look for the label tag. So there is many ways actually you can use it to track the nanoparticle, to quantify it, to visualize it. It depends on the settings and the type of the nanoparticle itself. Dr. Abdullah, I don't hear you, Dr. Abdullah.
Thank you. Is there any other questions from the audience?
Yes.
Comments, Dr. Azhar, please.
First, I want to make a small comment. You talk about Faraday. In fact, Faraday is the basis of Maxwell's equations. So there's no doubt about that he is the father of Maxwell's equations. That's clear. Second point is, we are talking about the very small thing in nanotechnology. Things are moving even further, and they're going beyond that into what they call the picotechnology. Picotechnology can be very rich because the quantum mechanics and its rules come up and make the things much richer. Can you say anything about what is happening towards the picotechnology?
Well, actually, Dr. Asghar, to be honest, I'm not expert or even have like really rich knowledge about the picotechnology. But what I know that it's related to maybe spectroscopy and analytical tools rather than a material itself. So we are not talking about like a material. We cannot synthesize material at the pico level, because pico level is approaching the diameter of the atom itself. If you look at the atom of gold is 160 picometer. Okay, so we are approaching the level of the atom itself. So we're going back to the physiochemical property of the atomic, which we know. We know how what is the oxidation state of gold, how they do react, what is the redox of that, how they assemble in lattice, what is the dimension of the lattice itself. So we know a lot about this. But to me, when we're talking about pico science, maybe we're talking about not a material dimension rather than a time scale dimension. Like if we remind the great work of rahimahullah, Professor Ahmed Zuwail. Ahmed Zuwail, he mastered the field of femtosecond spectroscopy and femtosecond imaging after that, okay? So this is my opinion, that the pico technology is, maybe Dr. Muthanna can help us here more. I think it's related to spectroscopy rather than a real material synthesis and application. I don't know if you agree or disagree also. This is like my best knowledge, but I don't claim that I have really deep experience in that.
Dr. Muthanna, do you have comment?
What Dr. Asghar is about. Dr. Asghar, it seems to me that in this case, going to the pico science, I would rather rely much more on the relativistic quantum mechanical picture of matter and in the pico scale, rather than trying to go inside that. Otherwise, you will have to go to the theoretical physics, which is the string theory and whatever it is beyond that. And this is for the chemical purposes. I think it's for the, as Dr. Alaaldin said, for the nanotechnology, I think it's of a minor advantage. Although I have the feeling, Dr. Alaaldin, that once you have supercomputers, you can calculate the biological properties of these medicaments and make predictions for them. And
Yes.
this will help a lot in the development of new drugs or new materials.
Yes, definitely. And there's a lot of artificial intelligence now and computer assist like understanding. Yes, yes, you're right.
Yes, yes. This is going to be the case, definitely. And I hope that one of the Arabic countries will be wise enough to buy a giant computer and make a center for this type of calculations. We need that.
Actually, thank you so much, Dr. Muthanna, for bringing this into the discussion table, which is the artificial intelligence, data mining, and computer assist modeling and simulation for pharmaceutical prediction and nano prediction. Dr. Muthanna, you know that all these things are used for even small molecule pharmaceuticals, not just for the nano.
Yes,
drug discovery nowadays is just driven by computers or mastered by computers. Unfortunately, and you mentioned that one of our universities, unfortunately, we cannot find like supercomputers in our countries, or even and even investment in this. And maybe Dr. Abdullah, things the University of Jordan were really seriously interested in is to invest in dry lab research as a startup now, because if you want to do a revolution in our scientific research, we cannot all of a sudden spend all these billions to build the infrastructure needed for nanotechnology or even for biotechnology or for any technology, old or new. It needs like millions or billions of dollars as infrastructure to catch up what is going on in the West. But the dry labs where you can generate amazing like math or amazing artificial intelligence based products or solutions or predictions. This does not require a lot of capital money to start up. We have a lot of brains. We have a lot of graduates. We have the smartest, maybe young people in the world. But we maybe we need to utilize them more efficiently and start more realistic. Like not looking for doing the mRNA technology. It's going to be really difficult to start up this business, for example, in any country of the Islamic world. But if you can manage to build up like thematic initiatives targeting like what, as Dr. Muthanna mentioned, artificial intelligence, data mining, simulation, drug discovery by computers, all these things, what you need is just fancy stations, supercomputers, and fancy brains and great management to these initiatives. So thank you, Dr. Muthanna, for bringing this to the table, because this is very important.
Dr. Alaaldin, please. Jordan has at the moment a functioning electron accelerator. Dr. Abdullah, what's the name of that?
Actually, yes, we have synchrotron.
Why shouldn't you be able to buy a supercomputer with the help of the European countries or maybe some of the United Nations?
Well, there was an attempt to have a supercomputer facility in Jordan at one time. That's probably about 6 to 10 years ago, but unfortunately it did not substantiate it into real thing.
In the future? In the future?
Maybe, yeah, maybe. I hope so, because they are thinking of it. But we have good capability in the pharmaceutical faculty at the pharmacy school, at
Dr.
University
Abdullah,
of Belgium.
this will reduce the efforts of synthesizing these nanomedicants enormously.
Yes, you're right.
You can't just just rely on trial and error in the experiment. You have to do this computative work just as well. And with the time, I think it pays.
I cannot agree with you more. Yeah, definitely.
Dr. Abdullah, Dr. Muthanna, I want to mention here that I'm now in Champaign. I'm now in Champaign, Urbana-Champaign, which is the University of Illinois. The second campus of it is in Chicago. In Chicago, the School of Pharmacy, the Professor Silverman, he's the discoverer of the gabapentin, the Lyrica, which is the drug selling like billions a year. The student who was working with him directly, okay, and like the most efficient student who helped him to do that is a Jordanian guy, you know. And as Dr. Muthanna mentioned, it was discovered based on computer-aided like algorithms and simulations. And this is an example how our brains can do in the West very great things, you know. But in our countries, we need to build the infrastructure. Back to Dr. Abdullah, comment regarding our experience in Jordan regarding the supercomputer. The supercomputer, to the best of my knowledge, Dr. Abdullah, as you mentioned, it was successfully initiated. Actually, it was initiated by orders
by His Majesty.
By His Majesty, yes, by His Majesty. Like, we want that, and we started that. But the problem that after we established the supercomputer facilities, we failed to maintain that. So we have problem is with the maintenance. Sometimes we can ask for European funds. Okay, no problem. We can get like building capacities, things, but this is not enough. You need like not just the foundation. You need the cruising stage. We need expert people to maintain, to keep it up, to keep, like, for example, electron microscope. We can buy electron microscope. We have in Jordan, like, for example, maybe 3 or 4 electron microscope at least. This is what I know, you know, maybe more even. But you cannot find an expert in electron microscopy in Jordan, because how much you're gonna pay him, this expert one, at the university level? All of us now, in Arabic university or in Islamic university, you know how much we can pay, like an electron microscope specialist. Like, you know, so we have a lot of things that we need a revolution, okay, to make it the whole package. It's not just getting the infrastructure. We need a system which can found the thing, cruise it, maintain it, keep it up, even develop it as function of time. So this is the problem we are facing.
Okay, is there any more questions or comments?
Can I ask you I want to thank Dr. Alkilany for his very interesting talk, and we were able to share our thoughts. It was a good moment for us. Thank you very much for your talk. We hope to see you sometime in the future, somewhere.
Thank you.
All the best.
Dr. Azra. Thank you, Doctor, and thank you for all the professors in the panel, our teachers. I appreciate this invitation again. Thank you so much.
Is there any other session?
تفضل دكتور يا مرحبا.
My concern is that you mentioned that we use liposomes as vehicle to deliver anti-cancer drugs, in which you have, of course, the nanotechnology thing. Okay, the selectivity of liposomes, to the best of my knowledge, does not guarantee enough level of attacking the cancer cells. It can attack any cell because it diffuses with the membrane of any cell. And as such, you are targeting all cells. It's so I don't know. I want you to elaborate a little bit more to clarify.
Thank you. Yes, yes. Thank you very much, Dr. Abdullah. This is actually a very important and interesting question, and I agree with you definitely that liposome is the electron microscope image of the duct cell I showed you. Confirmed the size of 100 nanometer, by the way, of the liposome, and you cannot reach 100% selectively that, okay, the liposome will keep going until it reaches the counter. It will end up there and not anywhere else. I agree with you. And to be honest with you, the preferred accumulation is not really outstanding even. We don't claim 100%, not even like outstanding. No, what they found in the clinical studies that there is not really preferential accumulation of the liposome, okay, in the tumor, but there is a decreased accumulation in the kidney itself, okay, in the kidney. Where is the nephrotoxicity happen? Where is the big side effect of the doxorubicin? So the conclusion, the clinical conclusion, that we are not gaining higher potency or selectively, but we are reducing the side effect, the serious side effect associated with the medication. And this is what the basis of the clinical approval of it. But here I don't want to stop, if you allow me to tell something. Okay, we didn't reach higher selectivity in the case of liposome or the doxorubicin. I want to give you two examples, two amazing examples, where the nanoparticles are smartly do the job in our body. Two clinically approved medicine. It's not a research paper or like a seminar. The first example, Doctor, the other liposomal formulation that I showed, which is the Amphotericin B Liposome. Okay, this Amphotericin B is anti-leishmania. It's an anti-leishmania drug. Okay, and we target the macrophage. So this parasite lives in the macrophage itself. The macrophage, you know, the macrophage. It is like a tank circulating in the blood. Okay, so we inject the Amphotericin B in the liposome, in a naked liposome, without polyethylene glycol. You know, the polyethylene glycol, which make it like a steel, which is we use it now in the mRNA vaccine. So this is not pegylated carrier, and we do non-pegylation for purpose, because we don't want it to circulate too much. No, we want it to be just immediately engulfed by the macrophage. And this is exactly what will happen immediately. So the macrophages will clear it immediately. So, and after that, the liposome will get into the site of action inside the macrophage. A very smart targeting. I'll give you another example. And remember, Dr. Abdullah, we did a lot of like business to prevent the macrophage non-specific uptake for the nanoparticle through make it seals, you know, through pegylating and many other strategies. But in this case, no, I don't want to make it pegylated. I want to get cleared immediately by the macrophages, because my enemy is inside the macrophage itself. And this is, I can claim, like one of the best targeting capability of nanomedicine. I want to give you a second example, because it's very important and very fast. If you remember, in one of the slides, I showed the free heme, which is the iron oxide nanoparticles. And if you ask anyone in the hospital in any country, they are using it, maybe in a different names. But if you ask about the sucrose iron injections, they will tell you, yes, we have it. And this is a nanoparticle. In fact, it's a nanoparticulate. If you put a droplet on a thermometer and you see it, you can confirm that it's a nano, or do dynamic light scattering. It's a nanopart. So why we use this? Why we don't inject just iron, soluble iron? Why iron particle capped with saccharose, stabilized with saccharose? Why? Because if you inject the same amount of iron dissolved in water to any patient, they're gonna die. It's too much iron in it. So what scientists managed to do is just make all this iron as a crystal at the nanoscale, capped with this like polysaccharide shell, and inject it into the patient. and immediately the macrophages again the macrophages will eat anything strange in our blood. So they will find it, will eat it, and what will happen after that? This gold core now inside the macrophage will start to itch and dissolve slowly, providing the body with a slow release of the iron to a level more time, but within the therapeutic level, without going with a spike with a very high concentration of the gold that it can really induce a lot of serious injuries or the toxicity. So these two examples demonstrate the targetability of the nanoparticles smartly to wherever you want to find a bug or to control the release of the matter. There is many other things like that. What I want to deliver, thank you, Dr. Abdullah, again for this question, because the cartoon which I showed that, this is a general principle, cannot be employed for every single modality of nanotechnology. No, like as I mentioned, nanotechnology is really broad field, and no one can claim that I am a nanotechnologist. When I introduce myself, I'm a nanotechnologist with a specialty in gold nanoparticle, even in gold nanorods, not gold like hollow cubes, not gold stars, it's gold nanoparticles. So every single class or subclass will definitely enjoy a pros or cons, advantage regarding to all attributes related to nanomaterials. Yeah.
Oh, thank you very much, Dr. Alaaldin, for this very enriching discussion of a very hot topic.
Yes.
And I would like really to thank all the audience and all participants for their fruitful intervention in the discussion. Thank you again, and I hope that we will see you again in our next session on nanoparticle by Dr. Yuldang Zaho, who is the Director General of the National Center for Nanoscience and Technology of China. This session will be held on Saturday, the 7th of August,
Inshallah.
2021 at 11 a.m. Amman time. Hope we'll see you again.
Inshallah,
Thank you very much.
All the best. All the best.
Thank you.
Shukran, Doctor. Thank you. Dr. Abdullah, before I leave, thank you so much. Thank you for this beautiful organization and academy for the invitation and host. And I would like to thank all professors for their questions, for their enlightenment. Really, I learned a lot through this fruitful discussion. And we'll see you again, definitely sure, inshallah.
Thank you. We'll see you in Amman very soon.
Keep